Welcome to the Wednesday afternoon lecture series. I'm Catherine Rogers, and I lead the unit on developmental signaling at NICHD. And I'm very happy to introduce our speaker for today, Dr. Mary Mullins. She's one of the leading experts in developmental patterning, and she is the vice chair of the Department of Cell and Developmental Biology at the University of Pennsylvania Perelman School of Medicine. And today she's the guest of the Women Scientist Advisors Committee. Mary did her Ph.D. with Dr. Gerald Rubin at UC Berkeley, and then she went on to do a postdoc in Tübingen, Germany with Dr. Janni Nusslein-Volhard. She was the first postdoc in Janni's lab to use zebrafish, and the goal was to recreate Janni and Eric Wieschaus's Nobel Prize-winning Drosophila mutagenesis screen, but this time in a vertebrate. And Mary was part of the team that really cemented the up-and-coming zebrafish model as a valuable research system. And Mary started her own lab at UPenn in 1995, and her research has drilled deep into the fascinating mechanisms underlying embryogenesis. She has determined how the distribution of signaling molecules is regulated. She has looked at how cells convert signaling levels and duration into gene expression. She has looked at how signal transduction, she's elucidated important signal transduction mechanisms, and she has also shown how maternally contributed factors in the oocyte influence early development. And Mary has influenced my own career, both scientifically and personally. When I was a postdoc in Tübingen, I encountered her at a conference, and she encouraged me to become a PI, and that turned out to be the final push that I needed to go for it. So she has very positively influenced the careers of many people, many scientists, including her lab members, her students at the celebrated Zebrafish Woods Hole course, where she's a faculty, and also during her tenure as president of the International Zebrafish Society. So today, Mary will tell us about the formation, function, and regulation of the BMP signaling gradient. And before I bring you on, I have been asked to announce the CME code one more time. Again, today's CME code is 44449. Okay, so now, Mary, welcome to NIH, and we're really excited to hear more about your research.
Thank you, Catherine, for that very lovely introduction. It's really a pleasure to be here. I always love coming to NIH. So much great science going on here, and I've had a really wonderful day so far, and I'm looking forward to tomorrow as well. So my title changed a little bit, and that's because I wanted to tell you about a new story, also involved in dorsoventral patterning, but not part of the BMP signaling pathway itself. It's part of the integrator complex, and I'll tell you more going on. So basically, my talk is divided into two parts. The first part, I'll talk about BMP morphogen gradient formation in dorsoventral axial patterning in the zebrafish, and the importance of some computational modeling that we've done that was really pivotal in changing our view of how this morphogen gradient is forming via some extracellular regulators. And then the second part will be about this subunit that we identified through some of our maternal effect screens called integrator subunit 6, and its role in modulating transcription to restrict the dorsal organizer in dorsoventral axial patterning. So one thing that we're very interested in is how morphogen gradients form. And morphogen gradients function by specifying different cell fates based on different concentrations. And so the shape of this gradient is really key to establishing the right proportion of cell types of these red, white, and blue cells shown here, as illustrated in these schematics. And the morphogen that we focus on is bone morphogenetic protein, BMP, and it plays a role as a morphogen in patterning multiple systems, in patterning the dorsal neural tube in vertebrates, patterning the digits in the limb in vertebrates, and a conserved function in patterning the dorsoventral embryonic axis. And this is conserved to Drosophila, as shown here, and this is a lovely picture from Ella Serpe's lab, actually, showing the peak of BMP signaling in the Drosophila embryo. So this is conserved from Drosophila to mammals, this early patterning. And this gradient of BMP is high ventrally, and I'll always have ventral on the left through the rest of the talk and dorsal to the right. It's high ventrally and low dorsally, and this gradient generates multiple sulfates across the dorsal-ventral axis, specifying the blood, the epidermis, the pronephros, the embryonic kidney, in a concentration-dependent manner, as well as the neural crest. And it also restricts dorsal sulfates to the dorsal half of the embryo, keeping neural tissue to this quadrant dorsally, as well as the somites. Through this screen that I did as a postdoc in Christiane Nusslein-Volhard's lab, we identified multiple regulators of the BMP signaling pathway. So on the left is wild type, and on the right is a BMP pathway null mutant. I think it's BMP7. You can see the elongated shape, of the embryo, and then you can see these somites here extending all the way around the circumference of the embryo rather than being restricted to the dorsal side as in wild type. Neural tissue is also expanding all the way around, and then it, sadly, bursts and dies due to that squeezing of the somites. And null alleles of all of these BMP pathway components exhibit that exact same phenotype. So they're playing a very important role. So the signaling pathway has been worked out by a lot of great work done by biochemists in cell culture, as well as in Xenopus from various different labs that we've greatly benefited from. From our mutant screen in the zebrafish, what we found is that a BMP2 and 7 are absolutely required for signaling. And through work I'm not going to tell you about, only this BMP27 heterodimer is able to signal in the embryo. And this is a part of my lab that's been working on this to try to understand the signaling mechanism for many years. And one thing that we've learned recently and published is that this heterodimer, it brings together two different type 1 receptors, ELK8 and ELK3 and 6. And what we've just learned recently is that the intracellular kinases of these receptors play specialized functions. So all the signaling just goes through one of those type 1 receptors, ELK8, and the other receptor we think is important in ligand binding. But I'm not going to talk about that today. And it suffices for you to know that the type 2 receptor, when the ligand binds this receptor complex, phosphorylates the type 1 receptor, activating it. And then it phosphorylates SMAD 158, and pretty much in the early zebrafish embryo, it's exclusively SMAD5. And then SMAD5 forms a complex, moves to the nucleus where it regulates gene expression. And an important readout that I'll be using is this phosphosmad5 for visualizing an activity gradient of BMP signaling across the embryo. So the ligand is regulated extracellularly by BMP antagonists, cordon, noggin, and follistatin, which can specifically bind the BMP ligand and prevent it from binding the receptor complex. And then cordon is negatively regulated by a metalloprotease called tolloid. There's another homolog called BMP1A, and I'll be telling you much more about that in a few minutes. And these metalloproteases specifically recognize cordon and cleave cordon, and cordon then releases the BMP to activate signaling. So these are negative regulators of the BMP antagonist cordon. And then there's another negative regulator, sizzled, which inhibits tolloid and BMP1A, and it binds the active site of these metalloproteases, blocking their signaling, or its ability to cleave cordon. There are additional extracellular regulators, twisted gastrulation and BMP-ER, which combine BMP and cordon in a tripartite complex, and they can both activate and inhibit BMP signaling depending on their levels. So there's a lot of extracellular regulation. There's been great biochemistry that's been done. But where these factors function in time and space is less well understood. And so that was part of what we wanted to understand. So the mechanism establishing the BMP signaling gradient has long been thought to be a counter gradient of cordon. So cordon is a secreted factor. It's expressed dorsally, and it's highly diffusive, and it was thought to form a gradient across the embryo and form a reciprocal gradient of BMP signaling. And BMP itself was thought not to be very diffusive. So that's also depicted in the schematic here with cordon on the dorsal side diffusing across the embryonic axis, and that generating a reciprocal gradient of BMP signaling. And this dotted turquoise line here is just where the BMP is expressed. So as I mentioned, this is conserved to Drosophila, and it's been well studied in Drosophila. And in Drosophila, it's known that the peak level of signaling here is generated by a shuttling mechanism. And a shuttling of whereby the cordon equivalent in Drosophila binds BMP, diffuses to this most ventral region, and toloid cleaves cordon, releasing the BMP, and that generates the peak level. And I'll show you a cartoon of how that works in a moment. So that's called shuttling. And it's basically a modified version of this counter gradient mechanism. It also has a counter gradient and then this additional shuttling. And there was some evidence in vertebrates that shuttling occurred, and this was from Naimai Barkai's lab, where her lab showed that in scaling in the Xenopus embryo that they had some evidence of this shuttling of the BMP. And then Eddie de Robertis' lab in patterning of the spinal cord had evidence to suggest that based on where BMP signaling was occurring versus where it's expressed and where cordon is expressed, that shuttling was occurring to generate the peak signaling level. So how would this work in the zebrafish embryo? So we have cordon here on the dorsal side, expressed here, it's secreted. These are the BMP heterodimer ligands, and this is here in purple, toloid or BMP1A, the metalloproteases. So the way this works is cordon diffuses, binds the BMP, it gets cleaved by toloid, releasing the BMP, and that generates higher BMP signaling levels there. So let me just show you that one more time in case you missed it. Cordon binds the BMP, diffuses, toloid cleaves cordon, releases the BMP, and generates peak levels of signaling. And so how can we test this? So what's very clear is from work in Drosophila, from Mike O'Connor's lab and Ella Serpe, that in Drosophila, if you knock out the cordon equivalent in Drosophila, SOG, if you mutate it, that you see a reduction in the peak level of signaling. However, if no shuttling is occurring, then we do not expect a reduction in the peak level of signaling. So this was the question: is shuttling occurring normally in dorsal ventral patterning? And the way we can test this is if we can develop a quantitative method to look at the signaling activity and measure it in wild type and in the cordon mutant. And this was done by a former graduate student in my lab, Joe Zinski, and he spent about a year working out a highly precise, reproducible method to visualize the BMP signaling gradient using a phosphosmad antibody. And so he would immunostain embryos with phosphosmad and co-stain them with some nuclear marker so he could identify every single nucleus in the embryo. And then he developed a program that would measure the phosphosmad immunofluorescent level in every single nucleus. And then we could convert that into a heat map shown here and visualize this signaling gradient. And then the idea is we can look at wild type at different time points, compare gradients between mutants and wild type as well. So basically we We can make these line graphs if we just look at the signaling level at the margin here around half of the embryo. So from, this is ventral to dorsal, and this is the gradient here shown, looking at multiple embryos. And if we want to combine embryos together, this is what the gradient looks in a single experiment in one set of embryos. So, okay, so does cordon shuttle the BMP? So what Joe simply did was immunostainings in wild-type embryos at different stages and cordon mutant embryos. And what you can see is that there's no reduction in phospho-SMAD levels. So, and in fact, it's what you can see, it's not reduced. It's also not increased, but it's expanded into the dorsal region in the cordon mutant. So cordon is clearly inhibiting BMP signaling in these lateral regions, but it is not playing a role in shuttling because we don't see an increase here or a reduction in the cordon mutant in the cordon mutant of phospho-SMAD. So why is there shuttling in Drosophila but not in zebrafish? So in Drosophila, you see a reduction of phospho-SMAD, and this was shown by Ellis Serpe's lab in the absence of cordon in Drosophila, but in zebrafish we do not see that. And we think it might be in part due to the different length and time scales during which this patterning is occurring. So in Drosophila, this phospho-SMAD gradient here occurs over a 30-minute time window. Gastrulation is very rapid in the Drosophila embryo, and the size of the embryo is much smaller as well. And so what we think is in the zebrafish embryo, which is much larger, gastrulation occurs over a much longer time period, that a gradient of signaling that's sufficient to pattern the embryo can be generated by possibly simple diffusion of either cordon or the BMP, whereas in Drosophila, this mechanism is required to shuttle the BMP to generate this peak signaling level. And Ella's lab has also shown there's some unique differences between toloid in Drosophila or cordon in Drosophila and in vertebrates, in that some of those differences in cordon can only be cleaved if it's bound to the BMP in Drosophila, whereas in vertebrates it can be cleaved whether it's bound to BMP or not. So that's also a key difference, and that plays a role also in generating these peak levels. Okay, so we know that shuttling is absolutely occurring in Drosophila. Our data indicates that it's not playing a role in patterning the dorsoventral axis in zebrafish. Another mechanism was proposed by Carolyn Hill's lab, where the BMP is expressed in a transcriptional gradient as well. And she had proposed that it's the transcription gradient that generates the gradient of activity in the embryo. And so we wanted to test that mechanism, as well as just generally establish other parameters that we could test this counter gradient mechanism. And we partnered with David Umulis and a postdoc in his lab, Shu Wang, at Purdue University to do computational modeling of the BMP signaling gradient. And this has turned out to be very powerful for us, and really a fantastic collaboration. So they developed these series of partial differential equations looking at the change in BMP, cordon, noggin, BMP and cordon complex, and BMP and noggin complex over time. And then they take into consideration the diffusivity of each of these factors, the binding constants, the decay constants, production, as well as toloid cleavage here. And they used these expression domains in the embryo for these components. And then the only assumptions, so the only data that we used, there's really good data from multiple labs about these binding constants. So we use the available binding constants in this modeling, but for all these other parameters, we decided not to make any assumptions and instead to just vary these parameters over 4 orders of magnitude. So we wanted the computer to tell us what fits, like what do these parameters have to be, which would allow us to do some tests of this gradient, these gradient mechanisms. So at the time that we did this, they did 1 million simulations and computer simulations, and it took like a couple months to do it. And this is just showing you, this is our wild type gradient. So then what we did is we asked which simulations fit our phosphosmad activity gradient. And this is just looking at wild type here. This is in blue, the actual gradient. And so those black dotted lines are solutions that fit. So in addition to looking at wild type, we also looked for solutions that fit our Corden mutant data, as well as Corden heterozygotes. And what we found of the 1 million simulations that were done, models produced, about 15,000 of them fit our wild type phosphosmad gradient, and only 4,000 of those fit both the Corden, additionally the Corden mutant and the Corden heterozygous data. And when we looked at the models, we were very surprised to find out that of these 4,000 models, only 263 of them were the counter gradient. So the counter gradient, as a reminder, Corden is diffusing across the embryo, generating the gradient of BMP. Most of the models did not show that. In fact, so we were perplexed, what are these other models? And it turns out that almost 3,800 of them were a source sink model. And no one in the field had really considered a source sink model, where in this case, Corden is a sink, and it's restricted to the dorsal side here. And it's basically removing all the BMP as it diffuses into the dorsal region, removing it from the system, and that BMP is diffusing across the axis on its own. So this, took a while for me to reconsider my decades-long view that a counter gradient was generating the Morphogen gradient. And so we went to test this further. And so part of the predictions from the model is that the source sink, shown in blue here, these are all simulations, and we're looking at BMP diffusivity. And in this model, BMP must be diffusive to diffuse and form the gradient. Whereas for the counter gradient models, it didn't really matter. Corden's the key thing. And in the field, I'm not, I guess, influenced by some data at other developmental stages, everyone thought that the BMP didn't diffuse. So we decided we should test diffusivity of BMP2. And this was done by Wei Bu in David Umulis's lab. And they basically did fluorescent recovery after photobleaching, FRAP. So using a BMP2 venus, expressing it, allowing it to diffuse, then bleaching it and measuring the time when the ligand diffuses back into this area. And so they measured it, and it was 4.3 microns per second. And basically, using that diffusivity, now it narrows down the models and reduces even further the counter gradient model, really pointing to the source sink model. So we've reconsidered now that the mechanism for generating the gradient may in fact be this source sink, but we really didn't eliminate the counter gradient. There's still simulations that fit for the counter gradient. So to test the model further, another graduate student came to the lab, Francesca Toison, and what was key in the simulations for the source sink model is that these metalloproteases played a really big role. And they basically prevented Corden from diffusing out into the ventral regions. So her thesis project was to test, do BMP1A. BMP1A and tolloid are homologous to each other, even though their names are very different. They're both metalloproteases. Are they restricting cordon's range in this source sink? So just a reminder, tolloid and BMP1A bind cordon, cleave it, and block it, and sizzled is an antagonist of those metalloproteases. And this is Francesca who did the work. She did a really hard experiment, which I'll tell you about in a moment. So she looked in a double mutant between BMP1A and tolloid, and indeed, the gradient is very depressed, as predicted, if they are functioning to restrict cordon to the dorsal side of the embryo. So in the absence of these metalloprotease, what we're seeing is cordon appears to diffuse across the entire axis and basically block all the BMP signaling. So, and we continued our computational modeling with the Humulus lab. And again, we looked at our, in addition to our wild type and cordon gradients, we looked at tolloid, BMP1A, and the BMP1A tolloid double mutant, and looked now, in this case, computer power was much better. Several years later, we could do 10 million simulations in like 3 weeks. And these in gray are simulations that fit the data. And now when we do the simulations, including these BMP1A and tolloid gradients, what we found is there are no solutions anymore that fit the counter gradient, and all the solutions that we did find were source sink models. So, and what's different here is, again, where is cordon? So for the counter gradient, cordon is going to diffuse across the embryo, but for the source sink, cordon is staying on the dorsal side of the embryo. And so these are the solutions that we found, and they all have cordon on this dorsal side. So to test this model further, what Francesca did was basically generate an immobile cordon. So if it only needs to be on the dorsal side to act as a sink, if we mobilize it in a region on the dorsal side, preventing it from diffusing, that should serve the purpose, and we wouldn't need the metalloproteases any longer. So, so that's what she set out to do. So she made a membrane-tethered cordon, and you know, she could show that this localized to the membrane. I'm not going to show you the data because it is published, and she could show that it functioned. It did inhibit BMP signaling. So, so this is the experiment that she did. She had a double mutant of BMP1A and cordon, and then injected a morpholino to knock down tolloid. And then she injected cordon, membrane-tethered cordon, and a lineage tracer into one blastomere at the 16 to 32 cell stage. And then she would look, in this cross, not all the embryos are mutant, so a quarter of them or a half of them, I forget how she did it, I think half of them, because she was able, anyway, half of them are wild type and half of them are mutant. So she, during the beginning of gastrulation, she would determine where her clone of cells were present. And then she would score the phenotype at 1 or 2 days post-fertilization, and then she would genotype. So everything's done completely blind. She doesn't know the genotype of the embryos until later. So these are some of her results where she restricted expression either dorsal or dorsal laterally. And these are the controls. This is the cordon mutant, also deficient in BMP1A and tolloid. And these are rescued embryos. And all of her data pointed to all her clones that were in this dorsolateral region were able to rescue. And sometimes you can see the asymmetry here where there's one eye that's smaller on one side compared to the other, and that's just because the clone was just on one side of the embryo. So let's see, I have to look at the time here. So basically, I hope that you'll consider in the future that rather than a counter gradient generating the BMP gradient in the embryo, which I think is still in the textbooks, in fact, what we think now is that it's this source sink mechanism. So, yeah, other things that we're interested in and have been studying, but I'm just going to briefly mention it, is how the gradient is interpreted, and the change in signaling levels from the peak level to this lateral region is about 4-fold phosphosmad level, so that's potentially the readout for the signaling. And so we're also interested in how this gradient gives rise to different gene expression domains that ultimately give rise to different cell fates. And a student in my lab has worked on this, and she's basically distinguished between these different morphogen interpretation mechanisms, either duration, slope, or concentration thresholds. And the targets that she's looked at seem to be regulated by concentration thresholds. And this is published, so I won't talk about it any further, except to say that Catherine has done some really beautiful work in this as well, using optogenetics, sophisticated optogenetics mechanisms to address some of these same questions. So the last part of my talk, I'm going to turn to this integrator subunit, 6, and its role in modulating transcription to restrict the dorsal organizer. And you have probably heard about the dorsal organizer that was discovered by Hilde Mangold and Hans Spemann in this dorsal region in an amphibian embryo. And what Hilde Mangold showed is that if she transplanted this dorsal organizer ectopically, ventrally, in another embryo, she could induce a secondary axis. So this is a major signaling center in the embryo that establishes the dorsal side of the embryo. And the organizer functions, chordin itself is expressed in the organizer, and of course the function of chordin is to inhibit BMP to allow dorsal development. And inhibitors in the organizer also inhibit Wnt8A signaling. And BMP and Wnt8A are very important in specifying ventrolateral cell fates in the embryo. But the organizer is inhibited itself, and there's feedback from Wnt8A through these transcription factors, VOX, VENT, and VED. And in fact, Igor David identified some of these, VENT and VED, I think, in in Xenopus and in zebrafish. And it's these transcriptional inhibitors that are important, acting through Wnt8 to repress the organizer so that the organizer doesn't extend into ventral regions. And actually, again, Igor David's lab showed another factor, LNX2B, which is expressed in the dorsal organizer that also inhibits the organizer itself. And I just mentioned the Wnt8 through VOX, VENT, and VED, and there's another factor, POW5F3, also functioning through VOX and VED, repressing the organizer ventrally. So in my lab, we've done maternal effect mutant screens, and one mutant that we identified is, we've cloned it now, and it's called INTA6. And basically, the organizer is hugely expanded in this mutant. So this is expression of goosecoid, which is in the dorsal organizer itself, in wild type. And what you can see in early gastrolith stage is the expansion of goosecoid all the way around the circumference of the embryo. And at later stages, what you see in these embryos is multiple dorsal axes forming. So rather than a single notochord here shown in wild type, you have these multiple notochords. And this is a unique loss of function phenotype in zebrafish. And you can induce multiple axes in many ways by ectopic expression of factors, but this is unique in the fact that it's a loss of function mutation. So as I mentioned, this is a maternal effect gene, and what that means is that the homozygous female is mutant, and she's putting an important gene product in the egg that's required for the embryo to develop. So if you cross her to a wild-type male, she produces 100% mutant embryos. So they're all heterozygous genotypically, but they all have a mutant phenotype. And this in contrast, if you cross two heterozygous INTA6 mutants to each other, all the embryos are completely wild-type. And you can raise the homozygous mutants to adults, and that's when you observe this maternal effect phenotype. And as I mentioned, this is because of maternal RNAs that are deposited into the egg during oogenesis, and these maternal factors are driving early embryonic development prior to the onset of zygotic transcription. And they also overlap with following the onset of zygotic transcription. So what is the integrator complex? It was originally identified by Ramin Shikater at the Wistar Institute next to Penn in Philadelphia. And his group showed that it's basically involved in processing snRNAs, that it cleaves off this 3' box of snRNAs, which allows them to become part of the spliceosome. The integrator is composed of these 15 different subunits, and they each have different functions, an endonuclease module, a backbone module, shoulder module. The INTA6 itself can bind PP2A, and that'll become important, the role it's playing, I think. And there's another related INTA6 called INTA6-like. And that's a conserved factor that's found in mammals as well. So there are numerous, in addition to this function in splicing and generating snRNAs, in the meantime, in the last 10 years, there have been numerous other mechanisms shown to be regulated by the integrator complex. Okay, so how does INTA6 repress the dorsal organizer? On a molecular level. It's either inhibiting dorsalizing genes or it's promoting ventralizing genes. So that's one question that William wanted to address. And then secondly, spatially in the embryo, where is it functioning? Is it functioning ventrally to keep the organizer to the dorsal side, or is it functioning in the organizer to keep it from expanding? And there are multiple components known to function either in repressing the organizer ventrally or in repressing the organizer and being expressed in the organizer itself. So William set out to try to address those questions. So the outline for the rest of the talk is shown here. I'll tell you about some of the genetics of this INTA6 allele, the original allele that we found, the spatial mechanism of INTA6 function, and then the possible molecular mechanisms for organizer repression. So INTA6, the original allele that we identified was a missense mutation. And so we wondered if it was a null allele or not. Maybe this is just, a hypomorph. So we requested from the Sanger Center some null alleles that they had and tested the phenotype. And so this is the INTA6 18A hub missense mutation where you see the multiple dorsal axes at 12 hours. And homozygous females of a nonsense allele die at a much earlier stage. So by 8 hours post-fertilization, they die. So clearly, the null allele has a earlier function, and it could indicate that there are actually two functions. And William could inject and rescue both of these phenotypes with wild-type INTES6 mRNA. And this is just a movie. Let's see, I think I have to. Of the different alleles. So this is the hypomorph, the multi-axis phenotype up here. This is the arrest, the null allele, and then these are wild types down here. And you can see the arrest ones arresting and then dying. There is a pause, so the mutant, hypomorphic mutants, they do pause at that same stage when the arrest mutants arrest. And many of them burst and die at a later stage when they have these multiple axes. So our original allele is a hypomorphic allele of INTES6. The mid-blastulus stage, when those embryos arrest, may be the first time that INTES6 functions. So what about spatially? Where is INTES6 functioning? So INTES6 mRNA is maternally expressed, so it's present everywhere in the embryo. So that doesn't give us any information. So William did something similar to what I just showed you, injecting INTES6 in just one cell at the 8-cell stage or 16-cell stage with a lineage tracer and determining, and he did that in an embryo that's transgenic, that contains a transgene, so it's the mutant embryo with a goosecoid GFP transgene, so he can identify the dorsal side of the embryo. And then he'd look at the beginning of gastrulation where these clones are present, and then look at the phenotype at 1 day, or a few hours later, 12 to 24 hours, and determine where does INTES6 have to be expressed to rescue the phenotype. So this is the INTES6 hypomorphic mutant with the goosecoid GFP, and this is one clone on the dorsal side of the embryo, showing the lineage tracer. And here's a different clone where, again, you have goosecoid GFP marking the dorsal side of the embryo, and then his clone on the ventral side of the embryo. And basically what he's found is that he can rescue the embryo when it's expressed ventrally, but not when it's expressed dorsally. And his data is all shown here. These are uninjected mutants. This is the key over here with the strong phenotypes in the blue and black. And then where the clone is expressed relative to the dorsal side, the organizer, and whether it rescues or not. And what you can see is that many of the clones on the ventral side rescue the mutant embryo. So that indicates to us that INTES6 functions ventrally, not dorsally, to repress the organizer. So what about the possible molecular functions? So I told you about already the first function that was identified for INTES6 in cleaving off the 3' box of snRNAs. There are two mechanisms by which integrator attenuates or represses gene expression. And one is a transcriptional attenuation mechanism identified by Jeremy Willits, actually at UPenn at the time. And in this case, short RNAs are cleaved and RNA polymerase is basically knocked off the gene complex in growing our mRNAs. So it's a way to reduce transcripts through this mechanism. Another mechanism is through transcriptional repression, where INTES6 and the integrator complex can basically bring PP2A to the C-terminal region of RNA polymerase and dephosphorylate it, basically repressing gene expression. And then there are a couple mechanisms where transcription is activated by cleaving enhancer RNAs, terminating them, or through cleavage of short paused polymerase, basically, where polymerase stalls on a gene, and so integrator comes along and cleaves the RNA and evicts the RNA polymerase off so that more polymerase can come along and transcribe at that locus. And then there's a role in DNA damage response. So the predictions for what to expect in the INTA6 mutant is if there's an snRNA processing defect, we would expect elongated snRNAs, along with a lot of unspliced mRNAs. For this transcriptional attenuation mechanism, we would expect many genes to be increased in expression in the mutant, and the same for the transcriptional repression. Genes that would normally be repressed would be expressed. For its activity in terminating eRNAs, we would expect longer eRNAs and a reduction in genes that are activated by these enhancers. And also for this POL2 pause release mechanism, we'd also see a decrease in gene expression. And at the maternal zygotic transition in the embryo, there's a DNA damage response that ensues. It doesn't require DNA damage, but some of the same components are used. And in that case, what we expect is an increase in maternal transcripts and a decrease in zygotic transcripts. So to look at this, what William did is he performed RNA-seq on the INTA6 mutant embryos to distinguish between these different possibilities. So he did a cross between a heterozygous INTA6 null allele to a male that's transheterozygous with the hypomorph over the null. And then he obtained sibling females, some that were just homozygous for the null allele, some that are transheterozygous for the hypomorph over the null that generate the multi-axis phenotype, as well as heterozygotes of the null allele, which have a wild-type phenotype. So we have these three different phenotypes. And then in addition, he did some true wild types, which there were no siblings he could use, but they're related but non-sibling females. So he crossed these homozygous these females here with the various genotypes to wild-type males. And then he collected from each clutch 25 embryos that he decoordinated and froze for RNA extraction. Another 6 sibling embryos that he fixed to precisely stage these embryos. And then an additional 25 embryos just to look at the phenotype, to see how strong the phenotype was. So he developed this method. He stained the fixed embryos to stage them. So these stages are very close in time that he looked at, all blastula, mid-blastula stages. And so to definitively know the stage, because the embryos look really similar to each other if you just look at them, he basically stained every embryo for nuclei and then counted them using a Fiji program so he could definitively count all the cells in every clutch. And then he kept those that were the right stage. He chose to do 3 stages. Let's see if I can get this pointer back. Okay. High stage, which is 1,600 cells, sphere stage, which is 3,500 cells, and then 20% epibole, close to what the zebrafish aficionados might know as dome stage, as 7,000 cells. And then we sent it off for sequencing. We used ribosomal RNA depletion, 150 base pair paired end reads, 40 million reads per sample, and normalized with these ERC spike-ins. And basically, I see I'm running a little bit late right now. Basically, what he found is a huge number of genes upregulated in the mutant, as you can see, at all of these stages, in either the trans-het or in the homozygous mutant. Most of the genes that are upregulated, shown here, all of these genes are upregulated, and shown in blue are genes that are normally not expressed in the embryo. So in wild type, they're not expressed, but they are expressed in this INTA6 mutant. And so what that tells us is that many of these upregulated genes are not normally expressed in wild type, and so that's consistent with this transcriptional repression mechanism with PP2A. And we think at these early stages, the chromatin is very open. There might be a lot of spontaneous transcription that occurs, and so this mechanism might be key to dampening all of that spurious transcription in the embryo. But what William's really interested in is how is this INTA6 repressing Wnt and or repressing the dorsal organizer. And so what he's found is that a lot of Wnt antagonists are upregulated in the mutant, both of the mutants. And so what he thinks is that repression, that INTA6 may be regulating Wnt8, and in the absence of INTA6, Wnt8 is depressed. Therefore, it's not repressing the organizer, and the organizer can expand in the embryo. And so our favorite mechanisms are these two, because of the large number of genes that are increased in expression, the transcriptional attenuation or repression by PP2A. But we can't exclude these other two because there are some genes that are downregulated as well. So I'll just finish by this last slide here, where these are known regulators that repress Wnt. The organizer here, POW5F3 is a direct transcriptional activator. VOX, VET, and VET are transcriptional repressors. Wnt8 is a ligand. LNX2B is part of a ubiquitin ligase. And INTA6 is yet another mechanism that we think inhibits an inhibitor to promote Wnt signaling, possibly by dephosphorylating RNA polymerase through the PP2A mechanism. And of course, INTA6 is also playing a major role outside of this in regulating zygotic genome activation broadly. So I'll just end with this last acknowledgement slide of the people in my lab. This is William Jones, who did all the work that I just told you about, Joe Zinski, who did the early modeling, and Francesca isn't shown here, but I also mentioned her work and her during when I told you about that. So thank you, and I would be happy to take questions.
Okay, thank you, Mary, so much for that really excellent talk. I think the INTS story is pretty berserk and very interesting. So my question is about, you showed in the mutant the expansion of the organizer around the entire base of the embryo, but it doesn't seem to expand into the animal pole. And I think you kind of touched on this towards the end of your talk, but how you showed it's required on the ventral side to keep it dorsal normally. But why does it not, in the absence of INTS, why does it not expand? Why does goosecoid not expand into the animal pole?
Yeah, I mean, it still requires nodal signaling.
Okay, so
and nodal isn't expanded. Yeah, so it does. It's still dependent on nodal signaling.
Okay, cool.
Yeah,
Thank you.
yeah, which is only at the margin of the embryo.
Really exciting talk. Thank you. You ruled out with your computational modeling what I guess, a transcriptional gradient, but I feel like I've looked at BMP stains on old-fashioned whole mount in situ, and it looks graded. I just wonder what you might have to say about how that grading is happening.
Yeah, no, it is absolutely in a gradient. Carolyn Hill first showed that. We've looked at it as well. There's definitely feedback. Its expression depends on BMP signaling itself at a certain point. So it might be regulated in that manner. FGF signaling early on is also playing a role transcriptionally in regulating it. So yes, I think, the expression domain is important, but it's only part of the story, So with the modeling, we can change the expression, and you can get, it still does diffuse, of course, in all directions. So, but absolutely, I'm sure that the transcriptional gradient is important. It's just part of the story. And that's evident because in the Corden mutant, the transcriptional domain doesn't change, but the gradient hugely expands. So it's only part of the story, yeah.
Thank you.
Thank you for that talk. Following up on Ben's question, I was wondering how much toloid expansion impinges upon the ability of Corden to function as a sink. Is toloid migrating into the dorsal part? Have you anchored toloids on the different sides to explore what it does to the gradient?
Yeah, so Corden, toloid, so BMP1A is the key of the two. So BMP1A is the more important one than toloid. And BMP1A is maternally expressed, and so it's everywhere.
Oh, okay.
Yeah, and toloid doesn't come on until early gastrulation, and then it is restricted ventrally in the embryo, and it's regulated by BMP signaling itself. But even in a BMP1A heterozygous embryo, the gradient is almost flat. It's crazy. And then it recovers when toloid gets expressed. But initially, it must be highly diffusive, absolutely. But we haven't done those experiments, and definitely those would be good ones to do. Yeah.
Thank you.
Hi, Mary. Great talk.
Thanks.
So I was curious about in the second part of the talk, where instead of getting a spot, you get a circle. And so that change in an organizer system from a spot to a ring, in theory, can be associated with a reduction in the height, the maximum level the organizer genes self-amplify. Mm-hmm. So if it saturates at a lower level, a spot-like pattern will turn into a stripe or a ring. So I was wondering whether in your analysis where you looked at all the different genes, whether many organizer-related genes were expressed but at a slightly lower level than they might normally, because that would be consistent with this explanation that I've seen.
Yeah, they're not less. They're not reduced. They're increased. So goosecoid is actually increased. Corden is up a little bit too.
Were they tested at the point at which you had multiple spots or when it was still a ring? Because at the point at which you have multiple spots, I would imagine that you would get an increase because it's like you have multiple organizers. Mm-hmm. But at the stage at which it's from a spot to a ring, that may be slightly different. I'm not sure.
Yeah, we see an upregulation, at the earliest stage is high stage, which is, not even an hour after zygotic genome activation. So it's 30 minutes later. So it's really early. And already we do see goosecoid up. It's on the dorsal side, so it fits with what you're saying, actually. The only thing is that we can express INT-S6 ventrally and completely rescue the phenotype.
Yeah, so that's related, I was wondering, to the diagram you showed where Wnt is feeding in to reduce, to inhibit things, and then the activity of the Wnt was getting exaggerated because you said it's playing as an inhibitor of an inhibitor.
Right.
So that could potentially explain it. We can talk about it later.
Yeah.
Okay, I have two questions online, if you don't mind.
Yes.
And we'll finish up. So our colleague Tom Schneider asks, are the proteins diffusing around the cell?
Yeah, I mean, we haven't looked at those proteins directly. So we've made the BMP venous fusion, but it's not expressed at physiological levels. So that's something that we're aiming to do, absolutely. It's harder, and I think we can only really visualize them with super resolution. You can't visualize them on a normal confocal. So we've tried to do those experiments, but the level, and we want to do it at a physiological level, so we just want to inject the amount that'll rescue, but we can't actually see the expression at that level. So, but I think with super resolution, we should be able to see that, and maybe we can we did that, some years ago. I think they're better fluorescent markers now that would be, make it possible to do that. But those are good questions, and that's definitely directions that we want to go in.
Okay, and then the final question for the future. Are there any clinical correlations or translational therapeutics related to your research?
Yeah, the heterodimer mechanism, I tell everybody that uses, making organoids or whatever you're doing, iPSCs, like if you use a BMP heterodimer, it's going to be much more effective than homodimers. I mean, there's data, not just from my lab, but from Jan Christian in mouse, that there are multiple biological contexts where the heterodimer is functioning. And in cell culture systems, it's been found by multiple groups that the heterodimer is always more effective. So I recommend heterodimers. You can buy them from R&D systems. They sell the homodimers as well as the heterodimers. So that would be, I would say, maybe some of the most direct impact.
Great. Okay, I think that's it.
Okay,
All
thank
right,
you very much.
Thank you very much.